During the next segment of the lecture, we're going to be talking about transport mechanisms. And what we want to do is follow the nephron from the proximal convoluted tubule to the descending limb, the loop of Henle, the ascending limb, the distal convoluted tubule, and even talk about the collecting duct and what happens there. And for all of these points of the nephron, what we're going to see is a peritubular capillary that is next to the tubule of the nephron. And so you'll have a lot of pictures that look like this one. And what we want to do is try to see the movement of material either from the tubule lumen towards the blood or from the blood to the tubule lumen. And so let's recall where we stopped. We stopped at glomerular filtration. And in the filtration part, what we noticed is that material just moved based on size, and that we lost a lot of the good stuff as we were getting rid of the bad stuff. And so the next step that we want to look at is a process known as reabsorption. And in reabsorption, we want to bring back all that good stuff that we lost: H2O, glucose, amino acids, sodium, other electrolytes, etc. And then if we want to get some stuff from the blood into the tubule, we don't call it filtration anymore because it's very selective, and so we call it secretion. And so these are the three processes that we see take place in the nephron, and we've already dealt with the filtration part of it, so now we have to look at the reabsorption and the secretion. So in this picture, what we notice is overall the material is going from the lumen, right, towards the bloodstream. You can see that that's the case for the sodium, and that's the case for the glucose. So this picture is showing us an example of reabsorption. And so let's look at how this works in more detail. So this is the same picture, and what we want to do is track what's happening. And if you recall, just to summarize, we've got blood that's going in to the afferent arteriole, will loop around this glomerulus, and stuff will start leaking out based on size, and we call that filtration. Now, this material is now known as filtrate. It's not urine yet, it is filtrate. And the reason why we don't call it urine is because it will change throughout the course of the nephron. But this filtrate basically is going to be going through this proximal convoluted tubule, and every step of the way there's going to be a peritubular capillary next door, and there's going to be some exchange that takes place. So let's just take this part right here of the proximal convoluted tubule in this box and see what's happening at that point. So here what you have is a set of cells that are lining this nephron. These are epithelial cells. They are, it's a simple cuboidal epithelium, and here are the cells in a zoomed in picture. So we zoom it in, and we see that these are the cells just in that box, and these are the cuboidal cells that are forming the epithelium of that tubule. So the lumen would be the space here, and this part of the cells is the apical surface, which will be making contact with the filtrate. And then this part right here would be the basal surface, and then you would have the peritubular capillary right here on the basal side of that epithelium. And so what we want to know is, well, if we're going to reabsorb the good stuff, and be selective about it, then how does that process work? So let's just say that we're looking at these two cells and we want to see how these two cells function. And so here is the close-up view of that. This right here is a drop of the filtrate that's moving downwards through this tubule. And this filtrate is going to have the good stuff and the bad, but really we're focusing on the good stuff. So H2O, sodium, amino acids, glucose, etc. And here we're going to track especially how, in general, we want to create an environment within these cells that is going to end up becoming an environment where absorption takes place and eventually reabsorption back into the bloodstream. So all these cells in the proximal convoluted tubule have a basal surface, and on that basal surface you have these sodium potassium pumps that are studded all over that basal lateral aspect of that cell. And these sodium potassium pumps, they work with ATP. So ATP is hydrolyzed. That's the energy where it gets hydrolyzed to ADP and PI, right? So ATP is adenosine triphosphate, and we're going to turn it into diphosphate and inorganic phosphate. And that energy is going to get the pump to push out from within the cell 3 sodiums, and it's going to bring in 2 potassiums. And cells in general are leaky to potassium, so a lot of this potassium is going to end up leaking out as soon as it's pumped back in. Really, we're going to end up focusing on the sodium. And we're going to use that sodium gradient to actually get these cells to do what they're supposed to do. So the result of the sodium-potassium pump, and again, we're just going to focus on the sodium here and kind of ignore the potassium, is that these cells, and I'm going to look at this cell now, which is doing the same thing, but I've simplified it. And this cell is going to be doing the same thing. It's going to be pumping out sodium, which means that the level of sodium within that cell becomes very low. So imagine that this cell now has very, very little sodium within it. That's going to create a gradient of a lot more sodium outside the cell, right? Outside the cell, there's going to be a lot more sodium than inside. And this is going to cause a type of drag force where whatever sodium is inside the filtrate will get dragged into the cell. And that's because it's going to be in higher concentration, right? These are simple laws of diffusion. There's going to be a higher concentration of sodium in the filtrate than inside the cell. And so the sodium will get dragged inside. Well, sodium is always associated with chloride, right? So also the chloride, which is going to stick to the sodium, will also get dragged with it. So this is just a very simple way of seeing how this sodium gradient works. Now, once that sodium enters the cell, we know what happens. There's going to be sodium-potassium pumps on the basal side, and the sodium will get pumped all the way to the bloodstream for reabsorption. So now take this idea and let's apply it to what happens with glucose. So we're in the same spot here. We are still looking at this particular segment of the proximal convoluted tubule. So it's the same picture. We're looking at these cells. There's going to be a filtrate here and a peritubular capillary there. And basically what we want to do is reabsorb material that we lost through the glomerular filtration. We want to bring it to the bloodstream. So what we're going to do is we're going to do the same thing that we did before. which is that we are going to have these sodium-potassium pumps that are going to pump a bunch of intracellular sodium out of the cell on the basal side, and it's going to end up going into the blood. And that's going to create a situation where the sodium levels that are within the cell drop significantly. The result of this is that the sodium levels that are outside in the tubule are much higher than the sodium levels that are inside. And so what you're going to have is an overall drag feel of sodium trying to go from the tubule lumen into that cell. Now notice we've added a few other items here. We do have a glucose transporter, and we're going to see how that works, and that's going to be on the basal side. And then we also have a sodium glucose symporter on the apical side. And that's because the whole purpose of this is actually glucose transport, but we're going to use that sodium gradient to help the glucose get pulled in. So a symporter is a protein channel, right? These are protein channels that we've drawn in. Both the sodium glucose symporter and the glucose transporter are protein channels that are embedded within the phospholipid bilayer of these cells. A symporter, the word sym meaning together, is a protein channel that brings two items through it simultaneously. And the two items that it's going to bring are going to be the sodium and the glucose. Now, specifically, it's two sodiums. So 2 sodiums per 1 glucose get dragged together through this symporter. So again, why is this happening? Sodium levels are low. And that means that this cell here has low sodium compared to the outside where sodium levels are high. So sodium is going to want to go from a higher concentration towards a lower concentration. Now, the way that it gets there is through this protein, through this symporter, but it has no choice regarding glucose. It is a symporter, which means it's built so that 2 items have to be brought through. And these 2 items are the sodium and the glucose simultaneously get brought into the cell. So all of a sudden, we not only have reabsorbed sodium, we have also reabsorbed glucose. And now glucose finds itself on the basal side, and on the basal side, it's going to be a glucose transporter. Remember, the sodium is going to get pumped out with that sodium-potassium pump, so it has a mechanism to make it to the bloodstream. But the glucose needs its own mechanism, and this one is a glucose transporter, right? So it's only specific to glucose. Only glucose will get through that, and glucose will end up finding itself within the bloodstream. So now you have reabsorbed the sugar that you lost through glomerular filtration. The same exact process is going to take place for a lot of other good things that we've lost through filtration. So we're not going to go over all of them, but most of them are going to be through this sodium gradient that we create. But just as another example, we're going to use amino acids as another example just to bring the point home. So again, we're looking at this part and these cells, and we've got the peritubular capillary next door. We are in the proximal convoluted tubule, and it could be anywhere in this proximal convoluted tubule, right? We're just looking at this one part of it over and over again. just to replicate the picture. But just remember, the whole tubule is going to have cells that do this. And these cells, again, are going to have sodium-potassium pumps, bringing the sodium out of that tubule epithelial cell. That's going to bring the sodium levels down. That means there's going to be higher sodium outside this cell, specifically in the lumen of the tubule within the filtrate. But that filtrate is also going to have other good stuff that we want. So, for example, there will be amino acids in that filtrate. So these amino acids now have to get reabsorbed. And it's going to be a similar mechanism. They're going to get dragged into the cell with the sodium that gets pulled in. The amino acids then get brought all the way into the peritubular capillary through their own mechanism, and the sodium-potassium pump will bring the sodium to the blood. And so now we've reabsorbed the amino acids via that sodium gradient that we created with the sodium-potassium pumps on the basal surface of these tubule cells. Now, in general, we always say that water follows salt. Or actually, not just salt, but we should say solutes to be more general about this. And so think about it this way. We are reabsorbing sodium. We are reabsorbing chloride. We are reabsorbing amino acids. We are reabsorbing glucose. So all of these end up being solutes. Now, water is going to want to follow these solutes. So water is not just going to stay in the tubule lumen, it's also going to move towards the tubule lumen all the way to the bloodstream. Now, the protein that we're going to use to get water across, because of course we have these phospholipid bilayers on these cells which make it difficult for water to cross, we're going to use a protein known as an aquaporin, and the word aqua meaning water, porin meaning basically hole or tunnel. And these aquaporins are going to allow for the water to flow from the filtrate to the inside of the cell and eventually to the bloodstream. So again, the solutes are going to be moving from inside the tubule lumen into the cell, and water is going to follow because water wants to dilute. Water always follows salt or solutes. So just imagine your sodium, not only your sodium, your glucose, your amino acids, your chloride, etc. All these solutes are getting reabsorbed. Water will go with them. And so you're going to have aquaporins on the apical surface. You'll also have aquaporins on the basal surface so that water eventually will make it from inside the cell to the bloodstream. If you recall the carbonic anhydrase equation from the respiration chapter, and this was how we transported carbon dioxide throughout the bloodstream, CO2 combines with H2O to give us carbonic acid, and carbonic acid splits into this acid H plus and a buffer which is called bicarbonate HCO3 minus. Now we call it a buffer because in general, if H plus levels rise, this bicarbonate right here will be able to neutralize those H plus levels. So in general, when you are doing metabolism, you tend to become acidic. If you think about your building blocks and you've got fatty acids, amino acids, nucleic acids, right? All of these tend to make your body more acidic when you're doing metabolism, when you're breaking down these molecules the acid starts getting released from them. And so in general, we tend towards acidosis. And the opposite of this would be to become alkaline, and that's known as alkalosis. Notice that your nephron is built more for acidosis. In other words, the default is going to be in order to get rid of acid and to reabsorb the bicarb. And that's just because of how human metabolism tends to make you more acidic. And so we need to reclaim or reabsorb the bicarb, but we typically don't want to reabsorb the H plus. We want to get rid of it. We want to excrete it. Unless, of course, we are becoming alkaline, then the opposite would take place. But the default is going to be in order to fix acidosis, where we really want to reabsorb that bicarb. And so notice that bicarb is going to get reabsorbed throughout the nephron. 80 to 90% of it gets reabsorbed in the proximal convoluted tubule, but you're also going to reabsorb it in the nephron loop and also in the distal convoluted tubule and in the collecting ducts. Now, in the distal convoluted tubule and collecting ducts, the cells can switch in order to fix not an acidic situation, but an alkaline situation. So we're going to see that later on, but this can change if you are alkaline. You do want to actually get rid of the bicarb and hold on to the H plus. But let's just focus on this acidosis condition and try to see how we can keep on holding on to the bicarb and get rid of the H plus. So the picture we want to look at from your textbook is one that looks like this. And what you want to see here, first of all, is just kind of, it does get very, very messy here. So you just want to look at the important parts of this. First of all, notice that you do have your sodium-potassium pump on the basal surface of that cell. And we're still in the proximal convoluted tubule here. You also end up seeing here that you have this bicarb transporter on the basal surface, and this will be where the bicarb can go through that basal side of the cell all the way to the bloodstream. Notice that you also have carbonic anhydrase, and we're going to see that it's located not only outside the cell on the apical surface, but also inside the cell. And so we're going to have to use it twice. Also notice that in general, what you're going to try to do is get rid of the H plus, which you can see it getting pushed out of the cell. And the CO2 that comes in as a gas form is going to combine with H2O to give you your carbonic acid. But when it splits inside the cell, the H plus will get pushed out towards the lumen, and the bicarb will instead get reabsorbed back into the bloodstream. So let's try to break it down one step at a time. This is a cyclical mechanism, so you can really start anywhere you like and follow the cycle. First, let's orient ourselves. Carbonic anhydrase inside the cell. Carbonic anhydrase outside the cell on the apical surface. And remember what carbonic anhydrase is doing. It's taking CO2 and water and giving us carbonic acid, and that splits into H plus and bicarb. But these arrows do go both ways. So we can actually do the opposite. We can take the H plus and the bicarb, fuse them together into a carbonic acid, and then we can end up generating our H2O and CO2. So we are going to use it in both directions. So again, this is a cyclical mechanism. We can start anywhere. I prefer to start with CO2, and specifically how CO2 basically diffuses. Remember, it's a gas, so it can diffuse easily through a phospholipid bilayer, but it's going to diffuse into this tubule cell. So now you have to imagine the CO2 is inside the cell. There's going to be carbonic anhydrase waiting for it. H2O also is found inside the cell. And remember the aquaporins. So we can't draw everything in every single picture, but you have to still remember the aquaporins are there, allowing for water to come into the cell. Now, that H2O and that CO2, with the help of carbonic anhydrase, are going to fuse together to give us carbonic acid. This carbonic acid will split into its two components. The bicarb wants to get reabsorbed. So that's where this bicarb transporter on the basal surface will come in handy. That bicarb goes through that portal all the way to the bloodstream, and now we have regained our blood plasma buffer. Remember, a buffer is something that helps to regulate our pH. And so now we have our blood plasma buffer back in the bloodstream. Remember that in general, because we tend to become acidic, what we want to do is we want to get rid of the H+. And so what we have on the apical surface is a sodium H+ antiporter. And here we have to remind ourselves that, again, we are going to use the sodium-potassium pump on the basal side to push the sodium out of that tubule cell, bringing the sodium levels within that cell low so that there is a gradient of sodium where the sodium outside the cell is higher than inside the cell. And what's going to happen is whatever sodium is outside in this tubule lumen is going to get drawn into the cell, but this is an antiporter. It's not a symporter. An antiporter means that when one molecule is going one way, another molecule gets switched and gets brought the other way. So a symporter means two entities go in the same direction through the protein channel. An antiporter, which is what we're looking at here, is that these two entities are going in opposite directions. So the sodium is going to go in to the cell, and at the same time, the H+, which we want to get rid of, gets pushed outside the cell. And so now we have this H+ outside this cell in the tubule lumen. They got pushed out of the antiporter. Now you want to imagine the bicarb that you may have lost through glomerular filtration coming down through that filtrate, and what you want to do is reabsorb this bicarb. So again, this is a cycle. We could have started anywhere. We could have started with this bicarb. We started with the CO2, but we're going to get back to the CO2 in a second. This bicarb is going to fuse with the H+. How does that happen? There's actually carbonic anhydrase on that apical surface of these tubule cells that's going to help with this. And this carbonic anhydrase is going to get these two entities to fuse together to make carbonic acid. But this is only going to be temporary because carbonic acid, and now notice that we are actually going in this direction, right? We are using the equation in the backwards direction of what we're used to. This carbonic acid will then give us our CO2, which is where we started this cycle, and the H2O. And the CO2 again, right, if we're looking further down, will then get brought in. And just to recap, carbonic anhydrase will take the H2O from within the cell, give us our carbonic acid. The bicarb gets reabsorbed. The H plus gets secreted out, and it fuses with whatever bicarb is inside the filtrate, giving us carbonic acid again, and we can do this all over again. So notice that this is a cycle, right? This will just keep going. It's a mechanism that keeps on in general. And just looking at the big picture here, right? Don't lose track of the big picture. If the details get confusing, at least know what the big picture is. The big picture is that in general, HCO3 minus, or bicarb, or our blood plasma buffer, gets brought back to the bloodstream, and H plus, or the acid that accumulates inside our bodies, gets secreted out into the tubule lumen. Now, in general, what we're going to see is that the descending limb and the ascending limb have opposite functions. And let's look at the ascending limb first. The ascending limb, in general, is going to be impermeable to water. So water does not cross that epithelium. However, it is famous for reabsorbing a bunch of salts, specifically sodium, chloride, potassium, but also calcium and magnesium. All of these get reabsorbed. However, we should say that not all of them make it to the bloodstream. So a lot of this material will actually accumulate inside the interstitial fluid that is found throughout this medulla of the kidney. So a lot of these salts will just stay here before they actually make it all the way to the bloodstream. And that's going to create a situation where the medulla of the kidney becomes very high in solute concentration. Or another way to think about it is the salts go up in concentration. And in general, it'll become more and more concentrated. And the term that we use for that is osmolarity. So the osmolarity of the medulla of the kidney rises as we get deeper into the medulla. Now, this will make more sense as we keep talking about this ascending and descending limb and how they work together in order to create this environment, so that we can also eventually fine-tune the concentration of the urine that ends up making its way out of the collecting ducts. So, what about the descending limb? The ascending limb is, and let's just orient ourselves real quick, right? This right here is the Bowman's capsule. So the filtrate's going this way. It's going through the proximal convoluted tubule. We talked about the proximal convoluted tubule being really important for most of the reabsorption that takes place. And there's more to talk about there, but we only have so much time, so we had to focus on the certain topics and just leave some topics out. Just realize that. Now we are in the descending limb, and the descending limb is not going to reabsorb salts. Instead, it's going to be leaky to water. So water is going to leak out of the descending limb. So as this filtrate is traveling down the descending limb, every step of the way, water is going to leak out. And no salt and just water. Okay? So just water, no salt. So we're making our way this way. Think about the concentration or the osmolarity of the filtrate at this point after it's lost all that water but held on to salts. And remember that osmolarity is the term that we use for how concentrated a solution is. Now we make this loop in this loop of Henle. We make our way back up the ascending limb, and the ascending limb instead is doing the opposite. It is getting rid of all of these salts. Your sodium, your potassium, your magnesium, your calcium, etc. All those salts are getting pushed out. And in general, right, we are increasing the osmolarity in this deeper part of the medulla of this kidney. And that's because of this ascending limb that's pushing all this salt out, and that salt accumulates in this deeper part of the medulla. Now, some of it, of course, will also get reabsorbed all the way to the bloodstream, but not all of it. A lot of it will stay within those that interstitial fluid of the medulla of the kidney. So just realize that this loop right here is built this way so that these two limbs can work together, because water follows salt. So as this salt is getting pushed out of the ascending limb, the water is coming out of the descending limb in order to dilute it. And so this bend of the nephron is not just there for no apparent reason. There's actually a functional reason, a physiological reason for this anatomy. And if there's anything to take away from an anatomy course, is that form and function are always going to be coupled. Now, it really helps to imagine a whole army of nephrons doing this, because looking at one nephron doesn't really give you the whole picture. So you have to imagine nephrons side by side. And so here I've drawn 5 of them. And of course, there are many more in a kidney. And this is a 2-dimensional drawing, so you also have to imagine nephrons in front and nephrons behind this picture to really get the sense of how this is all working. Because it's not just going to be one nephron doing this. It'll be an army of nephrons that are all working together. So for example, if I take this nephron here, the second one that I drew, and I look at its ascending limb, right? The filtrate is moving up this ascending limb. It's pushing the salt out. But what about the descending limb of this third nephron that I drew? Well, the descending limb of that third nephron I drew is going to be next door to the ascending limb of the second nephron. And so this salt that gets pushed out of the ascending limb of nephron 2 is going to draw the water from the descending limb of nephron 3. So water will go to dilute that salt. And this goes on, right? So you can look at the relationship between nephrons 3 and 4. So this is just a reminder that it's not just one nephron doing this, but it's the nephrons all together creating a salty environment with their ascending limbs and diluting that salty environment with their descending limbs. At the beginning of this chapter, we mentioned that the kidney has cortical nephrons, which are most of the nephrons that you find inside the kidney, and juxtamedullary nephrons, which are the ones that are in fewer numbers but are the ones that allow for the kidney to make concentrated urine. So let's see how this works. First of all, cortical nephrons, the Bowman's capsule is higher up in the cortex. The loop of Henle does not go deep into the medulla, so it kind of stays more superficial. With juxtamedullary nephrons, on the other hand, the Bowman's capsule is a bit lower in the cortex, but the loop of Henle dives very deep into the medulla, and it allows for the medulla to become more and more concentrated with solutes. Now, we should say that the word that we use for the level of solutes is osmolarity. That tells us how concentrated any solution is. And just to give you a starting place for osmolarity, a number that you should remember is that blood osmolarity is about 300 milliosmoles. So let's start our conversation with that number and then see how this concentration of 300 compares to the concentration of the extracellular fluid that you find in the medulla of the kidney. So what we should remember is that the ascending limb of the nephron is throwing salt out. And as it's throwing this salt out, the extracellular fluid of the kidney, specifically the medulla, becomes more and more concentrated. And that's why you see this number 350 as being more concentrated than, let's say, blood osmolarity. But then you see that it becomes even more concentrated the deeper you go into the medulla. So you go from 350 milliosmoles to 450 to 550, 650, all the way down to 900 milliosmoles in the medulla of the kidney, and that's due to the cortical nephron. With a juxtamedullary nephron, because you have a longer loop of Henle, and you can go deep into the medulla, what happens is, as this salt gets thrown out of this ascending limb, you go from 350, 450, 550, but you can go all the way down to 1200 milliosmoles, which is a very concentrated extracellular fluid that you find in the deepest parts of the medulla of kidneys. We're going to see that 1200 is what allows for urine to be very concentrated during times of dehydration. So let's see how this is going to play out. We're going to look at a cortical nephron, and we're going to see, starting with the afferent arteriole, that the blood that goes into this glomerulus is 300 milliosmoles of concentration. And this blood in the glomerulus, we are going to filter out things, and we're going to filter out a lot of stuff, the good and the bad, remember, but basically a lot of that plasma basically just leaks out. But that plasma is going to be the same in concentration as blood. After all, we are leaking out most of it. And so it's going to mirror the same osmolarity that you find inside blood itself. So the filtrate inside the Bowman's capsule will be 300 milliosmoles. Now what we have to do is bring this filtrate into the proximal convoluted tubule. So if you remember, the proximal convoluted tubule is all about reabsorption. And what we want to do is reabsorb the solutes that we've lost. So the sodiums and the glucose and the chlorides and the amino acids get reabsorbed. But remember that water follows. And if the solutes move out but water follows, what you're going to have is a situation where the osmolarity of the filtrate actually does not change. So really think about this. In order to change the osmolarity of the filtrate, we would either have to lose the solutes only or the water only. But if we're losing both, then that osmolarity inside the filtrate of the proximal convoluted tubule will not change because the solutes in the water are going to be equal in terms of how much we're losing. So we are going to the ends of the proximal convoluted tubule with an osmolarity of 300. Now, if you remember, the medulla of the kidney gets saltier once we hit the loopy part of this nephron, right? So where the descending limb begins, all the way down to the loop of Henle and the ascending limb, what we're going to see is an increase of osmolarity in the extracellular fluid of that medulla kidney. And so what we're going to see is that this 300, as it's moving down, is going to run into a medulla that's much more concentrated at 350. What's going to happen at that point when the 300 runs into the 350 is that the water from inside the tubule will go to dilute that higher osmolarity extracellular fluid. And that's exactly what we know is already the function of the descending limb. Is the descending limb is for water to leave the tubule. And so water basically is going to leak out and dilute that 350. As that happens, the osmolarity within the tubule rises to 350 because water has left the tubule, which means the osmolarity within the tubule gets higher. And then this 350 runs into the 450, which means water is going to also want to go from the 350 to the 450. But the 350 is now going to become more concentrated and become 450 within the tubule. The next thing that happens is the 450 runs into a saltier or more concentrated part of the medulla at 550. So again, the water is going to go from the more dilute solution to the more concentrated solution. It's going to go from the 450 to the 550, which means the fluid within the tubule is going to get more concentrated at 550. And this process will just keep going and going and going until we turn the loop. But at the loop of a cortical nephron, the extracellular fluid gets to a concentration of 900, which means that is the most concentrated that our filtrate can be once it turns the corner at the bottom of that loop of Henle. What we're going to see here as we go into the ascending limb is that this 900 milliosmoles concentration of the tubule fluid will change because the ascending limb is throwing salt out every step of the way. If you throw salt out, you're becoming more dilute. And if you're becoming more dilute, your osmolarity will decrease. And so from 900 milliosmoles, you'll see that it drops to 850 milliosmoles. Again, because the salt is being thrown out, more salt gets thrown out, so you go to 750, to 650, 550, 450, 350. And then notice around this time was where we saw that medulla that was increasing in osmolarity and becoming saltier as we dived deeper into the medulla. What happens at this point is that as this filtrate goes up, it just keeps throwing the salt out. And now we're getting to a very, very dilute concentration of the fluid within the tubule at 100 milliosmoles. So by the time we make our way to the distal convoluted tubule, the concentration of the tubule filtrate is very, very low. This fluid is very dilute at 100 milliosmoles. And now what we have to do is change that concentration to either dilute it more. If we are extremely hydrated, we want to dilute it even more to make a very dilute, clear urine, or we need to change that and concentrate it again in order to make a concentrated urine if we are dehydrated and we want to make a much more concentrated looking yellow urine. So here's just kind of a overall summary of what we've been looking at here. The big thing to take away at this point is that here we're at 300 to start. We start becoming more concentrated within that tubule as we make our way down the descending limb. And when we turn the corner, if we're in a cortical nephron, we're at 900. We're going to see that in a juxtamedullary nephron, because it goes even deeper, we're going to get to 1200. And then as we make our way back up, we start becoming dilute again. And when we get to the proximal convoluted tubule, we're at 100. And remember, all of this is possible because water is leaving the descending limb and salts are leaving the ascending limb. So this was the concept of making a dilute urine. We want to look here at this 100 osmolarity at the distal convoluted tubule. And now notice as we make our way into the collecting duct, and we're going to see the collecting duct is very important for this, the collecting duct can either dilute it even more to 90, 80, and all the way to 65. If you're very hydrated and you're making a lot of urine and it looks clear, then that osmolarity may be as low as 65 milliosmoles dilute urine. Or if we want to look at a juxtamedullary nephron, what we notice is that we see that the osmolarity within the tubule changes all the way to 1200 when we make that loop. And again, it's because we're going deeper into the medulla. As we make our way back up, however, we dilute it again and we get to 100. But if we are extremely dehydrated, we don't want to stay at 100. We want to concentrate it, right? So we want to reabsorb a lot of the water that's in that fluid so that as we make our way through that distal convoluted tubule, notice it's all about water getting pulled out of that filtrate. So we get to 200, to 300, to 400. And again, it's all about the water leaving that filtrate. And if you have created a situation where that juxtamedullary nephron goes really, really deep and makes that medulla very, very concentrated, you can keep on pulling water all the way until your filtrate on the inside gets as high as 1200 milliosmoles of urine. And this would be the urine that you're making when you're dehydrated. So you go and you urinate, but only a little bit comes out, and it's a dark yellow appearance of that urine. At that point, you know that your urine is probably approaching 1200 milliosmoles, and at that point, you know that you are severely dehydrated, and you need to go drink water in order to bring your homeostasis of fluids back to normal. Now, um, the distal convoluted tubule and the collecting duct is going to have two types of cells that we need to talk about. One type is the principal cell. The principal cell is going to be the one that is in highest numbers. That's why we call it principal cell. And again, here we're at the very end of the nephron, at the distal convoluted tubule, and also we're going to see the collecting duct has them as well. These principal cells are going to be sensitive to antidiuretic hormone and aldosterone. And this goes with the conversation of the production of either dilute or concentrated urine, because these hormones will change that concentration of the filtrate as it's moving through the distal convoluted tubule and the collecting ducts. The other type of cell that we have is one that regulates our pH, and those are known as intercalated cells. So these are fewer in number, and these are the ones that typically are going to want to, again, reabsorb the bicarb for us if we are tending to acidosis, which we usually do, and secrete the H+. However, make a note here that these can actually switch their roles if you are having a situation where you are going into alkalosis. The opposite will happen. They will secrete the bicarb and actually reabsorb the H plus in order to fix alkalosis. And so this is the part that we're talking about. So far, we have looked at filtration at the glomerulus and Bowman's capsule. We've looked at several ways of reabsorption at the proximal convoluted tubule. We've seen that the descending limb is going to be the one that throws the water out of the filtrate, and the filtrate is going to get more and more concentrated as it makes its way down. But when it loops back up, the ascending limb will throw the salt out, and so the salt is coming out of the ascending limb, making the filtrate much more dilute. And now we're in the very, very last stages here, and we're going into the distal convoluted tubule and the collecting duct. And so notice that the principal cells are going to be the ones that are higher in number, and these are going to be the ones that are going to be sensitive to the ADH and the aldosterone. And the intercalated cells are kind of sandwiched in between those. And those are going to be the ones that are going to be important for the pH homeostasis with bicarb and H plus, and regulating those amounts for us. So we're going to find these types of cells in the distal convoluted tubule and in the collecting duct. So both of these sections will have these types of cells within them. So just to recap what the intercalated cells are going to be doing, they will be, in general, in times of acidosis, trying to secrete the H plus and reabsorb the bicarb. However, during times of alkalosis, the opposite will happen. So, and we saw how all of this with CO2 happens in the proximal convoluted tubule and carbonic anhydrase. So we're not going to go over that again. Look at that segment of the lecture again if you want to see the details of this. But just look at the big picture here with H plus being secreted out and bicarb being reabsorbed, and just know that the opposite can happen. So if we have a situation of alkalosis, right, and not acidosis, then what we want to do is the opposite. We want to reabsorb the H plus, right? Because if we're alkaline, we are too basic. So we want to bring acid back into the bloodstream. And we want to expel or excrete the bicarb, which is the entity that is neutralizing the acid for us. And so just realize that this is one scenario, and one scenario is with acidosis, and this is another scenario, right, with alkalosis. And these intercalated cells can do both. So regarding the principal cells, you want to think about antidiuretic hormone, a water-soluble hormone that binds to its receptor on the phospholipid bilayer of these principal cells. And if you remember what water-soluble hormones do, remember that it's going to be a G protein coupled receptor. And so you're going to have that whole G protein cascade of adenylate cyclase and cyclic AMP taking place. But eventually, what you're going to be doing is you're going to be targeting the DNA and the RNA transcription, and then the protein, we need to get to translation. And which proteins are you going to make if you are sensitive to ADH or antidiuretic hormone? You're going to make the aquaporins. And what the aquaporins are going to do, both on the apical surface of these cells and on the basal surface of these cells, is that as this filtrate is moving down with, let's just make up some values here. Let's say that there is a osmolarity here of 700 milliosmoles, and this water now gets taken from that filtrate and gets brought into the cell and all the way into the bloodstream. Then after that has happened, after that reabsorption of water has taken place, then that filtrate, if it's lost water, it's going to be more concentrated, and it will be, for example, 800 milliosmoles. So you are conserving water, you're bringing it back into your bloodstream, and at the same time, you are concentrating that filtrate. And just remember that if you are talking about a juxtamedullary nephron with that medulla getting more and more concentrated, you could end up making this filtrate as concentrated as 1200 milliosmoles in the deepest parts of that medulla, because water is just going to keep on leaving that filtrate, and in the presence of aquaporins, this water gets reabsorbed much, much faster. And just remember what antidiuretic hormone means. It means that you are stopping the production of urine. You're stopping diuresis, anti-diuresis, right? And so this higher value of 1200 means that you're going to be urinating less volume with a more concentrated urine, a darker yellow urine. Now, aldosterone, if you remember, is a lipid-soluble hormone, and that's why this receptor in this picture is being depicted within the cell, because lipid-soluble hormones just basically go right through a phospholipid bilayer and find their receptor within the cell, sometimes within the nucleus of that cell. But eventually, you'll have gene transcription and translation. And what is the protein that we're going to make in this case? The protein is going to be sodium-potassium pumps and sodium channels. So aldosterone works on sodium. And so when we create these sodium-potassium pumps, that sodium gets pumped out of the basal surface of that cell. That brings the sodium levels within the cell very much down. Sodium decreases, and therefore the sodium wants to go through these sodium channels into the cell, but the water also wants to follow, and especially if we have aquaporins there from the production of antidiuretic hormone, we'll have more of this water being able to follow. The water is going to follow the salt, and it's going to follow the salt all the way to the bloodstream, and now we've reabsorbed the water with the salt. So just remember that aldosterone works on salt, antidiuretic hormone works on water directly with aquaporins, but both of these hormones will be there simultaneously in reality, so they're going to be synergistic with each other, In other words, one is going to help the other one work much more effectively. The last thing we want to talk about is the mechanism of urine excretion. How do the organs that are storing the urine and then have to expel the urine going to work with each other? So really what we're talking about is once we've made the urine and the urine goes through the ureters, it gets stored inside the bladder, and within the bladder we're going to have some baroreceptors in that bladder that feel the pressure of that fluid accumulating. And then what you have anatomically right at the end of that bladder is the internal urethral sphincter. And then further down, what you have is the external urethral sphincter. So the external urethral sphincter is voluntary, meaning that you have control of it. And so you can either contract it or relax it at will. But the internal urethral sphincter is involuntary, which means that it is your autonomic nervous system that is going to open it when the fluid in the bladder accumulates, and that's going to give you the sensation of feeling like you have to go and urinate. Now, you're going to be holding on to that urine with your external urethral sphincter. While the internal one is open, you're going to be trying to hold on to it with the external urethral sphincter until you actually make it all the way to the bathroom. And then you can relax that external urethral sphincter and actually allow for urination to take place. Because the urinary system and the reproductive system develop uh, side by side, they're also very much linked anatomically. And so what we see with the male urinary system, here what we have is the bladder, and you can see the urethra here with the internal sphincter going, then the urethra going through that prostate, and that's known as the prostatic urethra. And then you see that you have the external sphincter a bit further down right there. What you also notice with the urethra, if you look at the reproductive system of the male with the testes, is that the sperm that's going through the vas deferens will make its way, loop around the bladder, and make its way through that urethra at that point as well. And so the urethra is a passage tube for both urine and sperm in the male. In the female, what you notice is that you have the urinary bladder and you have the uterus right above. And notice how they sit with the uterus right on top. And this is why during pregnancy, extra pressure will be placed on that bladder, and so that the mother will feel like she has to go and urinate much more often, because it's not only the pressure from within, from the fluid, but also the pressure that's being put from the outside by the growing fetus. Now, in this case, when you look at the anatomy, you notice that the internal urethra is right there. The internal urethral sphincter, I should say, is right there, and the external is a bit further down, right in there. And you notice that the urethra is only a passage tube for urine, right? It's not associated with any reproductive tube of the female. Now, the storage reflex is the reflex that allows for you to store volumes of fluid inside your bladder. Now, you have to remember that the bladder is a muscular organ. And so, as a muscular organ, it will contract if it needs to in order to expel the urine. So, the storage reflex is a continuous sympathetic stimulation. So, in other words, under sympathetic tone, the muscle of the bladder, known as the detrusor muscle, is going to be relaxed, right? So we're talking about the muscle that is the muscle that is making up the bladder there, called the detrusor muscle, is relaxed. If it contracts, it's going to push the fluid that is being stored within it out. So if we want to store it, we want to relax the detrusor, and that's from sympathetic stimulation. And your sympathetic nervous system, if you recall, is coming out of the center of your spinal cord. And so these nerves that come out from the center of the spinal cord go uh, and and give the signal to that detrusor to stay relaxed under sympathetic tone and give you most of the time this storage reflex. The second thing that we want to do with this continuous sympathetic stimulation from the center of the spinal cord is that we want to keep the internal urethral sphincter contracted so that it's closed off right here at that point. That's the internal urethral sphincter. Notice there's a branch from this nerve that also goes to the sphincter and basically keeps it contracted. So while the detrusor muscle is being relaxed in order not to push the fluid out, the internal sphincter is being contracted. In order to urinate, we go from the storage reflex to the micturition reflex. So that just means urination. And so this would be where the bladder fills up with fluid, and this distended bladder with its baroreceptors sends the signal to the central nervous system. And this is going to be through these sensory axons from those baroreceptors to the central nervous system. And what's going to happen is that the central nervous system will then switch from the storage reflex to the micturition reflex. And really what we want to do is the opposite. And so notice that at this point, it's going to be nerves that are further down in the spinal cord that are going to send signals to contract the detrusor, and that's going to push the fluid out. And at the same time, we need to relax the internal urethral sphincter. Now, this is all going to be autonomic, and so you're not going to have control over either of these things. They're going to be happening. That's going to give you the sensation that you feel like you need to urinate. At that point, you hopefully will have control of the external urethral sphincter. And once you relax the external urethral sphincter, the urine will be able to flow out of the urethra.